Punnett squares. The implications of Mendel's work are made easily visible by the work done by Reginald Punnett in the early 1900s. He extended Mendel's work and developed a schematic approach to predicting the outcomes of breeding. The probabilities of the genotypes and phenotypes of offspring resulting from the breeding of parents, given their genotypes, is simple to calculate for single traits. Punnett developed a tool, now called the Punnett Square, to determine the possible outcomes given the alleles of each parent. The Punnett Square relies on Mendel's law of segregation, where a parent can contribute only one of two possible alleles to an offspring. To set up a Punnett Square, first, the possible alleles that each parent can contribute must be determined. Every parent can only contribute one allele of each gene to each offspring because gametes are haploid. When making gametes through the process of meiosis, parents distribute one of their alleles to each haploid gamete. 50% of their gametes will have one allele and 50% will have the other. So a parent who has both alleles, capital A, can contribute capital A or capital A to each gamete, blue or red. A parent who is blue capital A, red lowercase a, will contribute either the capital A or the lowercase a. Lowercase a, capital A can contribute either the blue lowercase a or the capital A. I guess it's black. And if it's lowercase a, black, lowercase a, red, that parent will contribute either the black lowercase a or the red lowercase a. Black represents one parent's allele, and red is the allele from the other parent. A Punnett square is divided into four equal-sized smaller squares, shown here. The alleles of one parent, the two letters, are written on top, up here, and two letters for the alleles of the other parent are written along the side here. For example, write the alleles for a true breeding plant with purple flowers on top, where the two alleles are capital P, and for a true breeding plant with white flowers down the side, and that's lowercase p, lowercase p. Now, fill in each square with one letter from the box above and one letter from the side. We usually write the capital letter first. So you can see here we have capital P, lowercase p. I've shown you the first box. You do the rest, and I'll show you the result on the next slide. So we can see every box has a capital P from this parent, capital P, capital P, capital P, capital P, and a lowercase p from the second parent, lowercase, 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 lowercase. If fertilization results in the random combination of gametes, then each square represents a 25% chance of that genotype being the genotype of the resulting offspring. Since capital P is dominant, what will be the distribution of flower colors of these offspring? The inside of the Punnett square, so these represents a prediction about the F1 generation. And remember that capital B, capital P is purple. And lowercase p is white. All of these offspring will have purple flowers since capital P is dominant and appears in each box. The only way to get a white flowering plant is for capital P to be absent. Now, let's crossbreed two of the offspring from the previous page together using a Punnett square to get the F2 generation. In this case, both parents are capital P, lowercase p. We're just taking Two of the offspring, all four of those offspring in the F1 generation were capital P, lowercase p, right? Heterozygous dominant for purple flowers. So I've gotten you started here. You fill in the square and check your answer on the next page. This is the first step, just writing the two parents and their possible alleles on the top and on the side. Now fill in the boxes. All right, let's make sure we are all in agreement for our boxes here. So capital P from this parent, capital P from this parent in the first box, capital P, lowercase p, capital P, lowercase p, and then lowercase p, lowercase p. What will be the probability of each possible genotype and phenotype in the offspring? Here is how we find the genotypic ratio. Here are the genotypes in the four boxes. Capital P, capital P, capital P lowercase, capital P lowercase p, lowercase p, lowercase p. Each appearance represents a 25% chance of that genotype occurring in the offspring. So the probabilities are 25% homozygous dominant, 50% heterozygous dominant, and 25% homozygous recessive. This can be written as the genotypic ratio 1 to 2 to 1, where the homozygous dominant is 1, the heterozygous dominant is 2, and the homozygous recessive is 1. Let's look at the phenotypic ratio now and see how it differs a bit from the genotypic ratio. Anytime capital P is in the genotype, the plant's flowers will be purple. The only genotype that leads to white flowers is the homozygous recessive lowercase p that we see right here. That means that there is a 75% probability of offspring having purple flowers and a 25% chance of offspring having white flowers. The phenotypic ratio then is 3 to 1, where 3 is the number of purple flowers relative to the number of white flowers, 1. These are predictions, not absolute measurements. For this cross, it's expected that the ratio of purple to white flowers will be about 3 to 1. In the flower color example, this homozygous dominant and heterozygous dominant genotype plants have the same phenotype, purple flowers, but different genotypes. So we get purple flowers, whether it's homozygous dominant or heterozygous dominant. A phenotype is the observable characteristic, the purple flower. The genotype is the organism's set of genes. The above 3 genotypes result in purple flowers since the purple flower allele, which we've labeled capital P, is dominant. In this flower color example, the homozygous recessive lowercase p plant has white flowers. This is the only genotype which results in white flowers because the lowercase p is the allele that we've given for white flowers and is recessive. Example 1, Punnett Square. If a homozygous tall pea plant is crossed with a heterozygous tall pea plant, what percentage of the offspring will be tall? Tall is given by the allele capital T and is dominant to short, which is given by the allele lowercase t. So I'm going to work through it on this slide, and then I'll just summarize the answer on the next slide. So we're crossing a homozygous tall, which means both alleles are for tall, and a heterozygous tall, which means one of our alleles is for tall and one is for short. Okay, so let's look at the offspring we would have. Okay, so there's our Punnett square. And then let's Let's look at our genotype and phenotype percentages. So in our genotypes, we have two different ones. We have half of them homozygous tall and half of them heterozygous, but this is dominant, right? Okay. And so that means the phenotype will be 100% tall. This slide is a summary of the work we did on the previous slide, where we showed that in answer to this question, what percentage of the offspring will be tall? All 100 of them will be tall. Example 2, Punnett square. If a heterozygous yellow seed plant, so heterozygous yellow seed plant, is crossed with another heterozygous pea plant, yellow pea plant, what percentage of the offspring are predicted to have yellow seeds? So yellow seed, capital Y, is dominant to green seed, lowercase y. So we have one heterozygous plant crossed with a second heterozygous plant. So what do we get? Let's see. Okay, so I made a mistake. Anybody catch it? So we have 25% homozygous dominant. That will be yellow, right? We have 50% heterozygous dominant. That will also be yellow. And 25% homozygous recessive, and that will be green. So we would expect 75% of the plants to be yellow and 25% of the plants to be green or have green seeds. This is the seeds. So what percentage of the offspring are predicted to have yellow seeds is the question. And the answer is 75%. So this slide summarizes everything that we talked about on the previous slide. But again, just to answer the question, what percentage of the offspring are predicted to have yellow seeds? 75% are predicted to have yellow seeds. Test cross. A test cross involves breeding the individual whose genotype we are trying to determine with a homozygous recessive individual. If any offspring display the recessive phenotype, the parent must be heterozygous. So I don't know, this is a purple flower, but I don't know if it's homozygous dominant or heterozygous. So I cross-fertilize it with something I know is recessive, homozygous recessive. If this unknown purple flower is homozygous dominant, then all of the offspring will be purple because all of the offspring will have a dominant allele. If the purple flower is heterozygous dominant, then half of the offspring will be purple because only half will have this dominant allele, and the other half will be white because they will be homozygous recessive. Just because an allele is dominant does not mean that it is more common than its recessive counterpart in a population. For example, 1 out of 400 babies in the United States is born with extra fingers or toes, polydactylism. The allele for this unusual trait is dominant to the allele for the more common trait of 5 digits per appendage. In this example, the recessive allele is far more prevalent than the dominant allele in the population.